Very good evening to everyone. Welcome to this NPTEL live TS session for the course Cell Culture Technologies taught by Professor Mainak Das from IIT Kanpur. My name is Ankita De and I am a Prime Minister's Research Fellow pursuing PhD in Cancer Biology at the School of Medical Science and Technology, IIT Kharagpur. And I'm going to be one of the TAs for this course this year. I'll be hosting this live TA session every week on Tuesdays 7 to 8 p.m. And during this 1 hour TA session, we will be solving previous year's assignment questions of the year 2021 live. For any doubts or clarifications regarding the solution, you may use the chat box option to post your queries and we will try to solve or address them together. Towards the end of each session, we will have a Q&A segment where you can post your questions and we will discuss them. Further, we will also provide you with the course materials and class recordings for each session for access later and the link to Google Drive folder and the YouTube videos. So let's get started. Question number 1. Cells that are dash need to be cultured while attached on a solid or semi-solid substrate. So I repeat the questions. Cell that are dash need to be cultured while attached on a solid or semi-solid substrate. The options given here are living, non-living, anchorage dependent, and anchorage independent. So you may write the answers in the chat box and we will go through each one of them. So let's see what the correct option for this question is. So the right option is anchorage dependent. That is, cells that are anchorage dependent need to be cultured while attached on a solid or a semi-solid substrate. Now we will see why and how. So basically, anchorage dependent cells are those cells which require a surface for normal proliferation. Examples of anchorage dependent cells include the cells which are the normal tissue derived cells, whereas anchorage independent cells are those which do not require a surface for their growth. They usually go in suspension, and the examples are cells derived from the hematopoietic system or transform cells. Further, dead cells too do not remain attached to the surface. Once live cells undergo growth arrest, they detach themselves from the substratum. So therefore, the right option is anchorage dependent cells. Question number 2. So which of these chemicals is used as a protective agent when cells are stored frozen? The options are only DMSO or dimethyl sulfoxide, only glycerol, uh, option C, both DMSO and glycerol, or option D, none of DMSO and glycerol. So let's see what the correct option should be. So the right answer for this question is both DMSO and glycerol. So both DMSO or dimethyl sulfoxide and glycerol are used as cryoprotective agents for the purpose of cryopreservation or the freezing of cells. So first and foremost, we need to discuss what is the point of preserving cells. So as we already know that cells cannot be kept in culture continuously. So therefore cryopreservation is the method that comes into play here. It is the technique by which cells can be preserved for long-term storage at sub-zero temperature, usually at minus 196 degrees centigrade. For this purpose, we use appropriate freezing cryoprotective agents for the purpose of cryopreservation. They help to bring down the melting point, the freezing point of the of the medium in which cells are stored. Also, they allow a slower rate of cooling to prevent the formation of intracellular or extracellular crystals during the process of freezing. Now let's move on to question number 3. Question number 3 is also somewhat similar to question number 2. So the cryopreservation of cells is done at temperatures. The options are below 0 degree centigrade, below minus 130 degree centigrade, above 0 degree centigrade, or above 100 degree centigrade. Now let's discuss the right answer. So as we already discussed in the previous question that cryopreservation is the technique by which cells are stored at sub-zero temperature for long-term storage. The sub-zero temperature is usually maintained at minus 130 degree centigrade. So minus 130 degree centigrade is the right answer. In For this purpose, we usually use electric freezers or liquid or the vapor phase of nitrogen, and the temperature that is usually uh, attained with the help of the liquid or the vapor phase of nitrogen is minus 196 degree centigrade. Question number 4. Aseptic techniques are required to prevent contamination in cell culture, to ensure contamination of cell cultures, to ensure non-sterility of cell culture, or to prevent only fungal attack. I repeat the question, aseptic techniques are required to prevent contamination in cell culture, to ensure contamination of cell cultures, to ensure non-sterility of cell culture, or to prevent only fungal attack. Now we will discuss each of the options here and find out the correct option. So, aseptic techniques usually refer to the techniques which prevent the contamination of our cell culture from microorganisms such as bacteria, fungi, and viruses. Therefore, we should maintain proper sterility condition during the culture of cells. For this purpose, we need to keep all our equipments and media or reagents which are used for the culture of cell culture entirely clean or free of microorganisms such as bacteria, viruses, and fungi. Dirty work surfaces or unclean incubators are the source of biological contamination. Aseptic techniques are therefore designed to act as a barrier between the microorganisms in the environment and the cell culture, and it depends upon a number of procedures to reduce the probability of contamination from these sources. The elements of aseptic techniques are a sterile work area, good personal hygiene, sterile reagents and media, and sterile handling. So, in order to prevent contamination in cell culture, we need to follow a set of rules and procedure. We need to keep our work surface entirely clean. We should repeatedly clean the surfaces with 70% alcohol to ensure that all microorganisms are completely wiped off before working working on our cell culture before working on our cell culture plates. Also, the cell culture room must be entirely free of all kinds of contamination. It should be regularly cleaned to ensure that no source of biological contamination is present. So these are some of the techniques by which we can ensure that our cell culture is completely free of microorganisms. Now let's move on to the next question. Cells prefer to attach on hydrophobic surfaces, hydrophilic surfaces, fluorinated surfaces, or all of the above. I repeat the question, cells prefer to attach on hydrophobic surfaces, hydrophilic surfaces, fluorinated surfaces, or all of the above. So the right option is hydrophilic surfaces. So we know that during the in vitro culture of cells, tissue culture plates are usually used. Cells attach to the surface of the tissue culture plates for proliferation, as we have discussed previously, that anchorage dependent cells attached to the surface of the tissue culture plate bind to them and grow. However, cells do not attach directly to the surface of the tissue culture plate. They instead bind to the poly-L lysine coating, which is with the help of integrin molecules present to the surface of the cell. Tissue culture plates are usually coated with a coating of poly-L lysine, which helps to create a positive charge on the surface of the tissue culture plate. The surface of the cells are usually negatively charged, which helps these cells to bind to the surface of the tissue culture plate and enable them to grow. Integrins are the molecules which help in anchoring the cells to the tissue culture plate. So the cells prefer to attach on hydrophilic surfaces. So what are which of the following molecules that are used for surface coating for cell attachment is of organic origin? The options are aminosilin, poly-L-ornithine, poly-D-lysine, or collagen. So we have previously already discussed that poly-L-lysine is used for coating the surface of the cell culture plate, and it helps cells to bind to the surface of the cell culture plate. So definitely poly-D-lysine is not the correct option. Now we also need to find from amongst these options which of these following molecules is organic in nature. Now let's find out the right answer. So collagen is the right option. So collagen is also one of the structural proteins which is found in the extracellular matrix of cells. Extracellular matrix refers to the mm, external environment that surrounds the cells. Collagen is often used to coat the surface of tissue culture plate. Apart from polyl-lysine, is basically composed of three chains which is wound together in a tight triple helix structure. The structure of the collagen triple helix is shown in the adjoining figure, where we see three chains tightly wound around each other, and each of these chains are composed of three consecutive amino acids: uh, glycine, hydroxyproline, and proline. Glycine helps the chains to tightly wound around each other due to its small size. Hydroxyproline and proline are the two other amino acids present. Now let's move on to question number 7. Cell culture media by the name of DMEM stands for Dalbecko's modified eagle medium, dermal mesenchymal epithelial medium, Desmin modified epidermal medium, or the dual molecule expansion medium. Now let's find out the right option for this question. The cell culture medium by the name of DMEM stands for Dalbecko's modified eagle medium. This is a widely used basal medium used for supporting the growth of many different mammalian cells. Now let's move on to the next question. Question number 8. Phenol red indicator is primarily used in cell culture medium for temperature indication, pH indication, osmolality indication, or freezing point depression indication. Now let's discuss the correct option. So phenol red indicator is a pH indicator that is used in cell biology laboratory. Its color gradually transitions from yellow to red over the pH range of 6.8 to 8.2. That is, it shows a yellow color when The pH is more towards the acidic side, whereas it turns to pink when the pH is more towards the alkaline side. It helps to indicate the condition of the cell culture medium, and it indicates whether the cell culture medium needs to go through a media change or there are any kind of contamination present in our cell culture. So this is mainly indicated by the following, that is waste products which are normally produced by dying cells or overgrowth, or an overgrowth of contaminants often lead to the formation of an acidic condition in the cell culture medium. And this leads to a change in the color of the phenol red indicator, that is, it turns more yellow over time. This indicates that there might be some kind of contamination from bacteria present in the media, which means that we need to uh, discard that particular cell culture, and or it could also indicate that waste products are being produced in the cell culture and we need to change the media of the cell. Mammalian cell waste products themselves also slowly decrease the pH, gradually turning the solution more yellow over time. This indicates that the media shall need replacing in the absence of contamination. Let's move on to question number 9. Which of these cell culture media is carbon dioxide independent? DMEM, MTESR, L15, or NBA active 4? So let's see what the correct option is. The L15 medium, the Leibovitz is medium, is used to grow cell line in the absence of CO2. So normally cell culture media need some kind of buffering with of CO2 to survive. It is, we need to culture the cells in the presence of 37 degree centigrade temperature and 5% CO2 condition for its proper growth. For that purpose, we need to add sodium bicarbonate to the medium, which helps in the buffering of the of CO2. However, this particular medium, that is the L15 medium, do not have this kind of a requirement. It is used to culture cells in the absence of in the absence of sodium bicarbonate CO2 buffer system, and this it does by using the free basic amino acids phosphate buffer and higher levels of galactose and sodium pyruvate present in the medium. And this also obliterates the need to culture the cells in presence of CO2. Can be cultured under conditions of free gaseous oxygen with the atmosphere. Question number 10. Fetal bovine serum or FPS is extracted from fetal calves. Which of the following statements is true about FPS? That it contains cells, it contains fibrin, it contains clotting factors, or it contains growth factors. So the right option is FPS contains growth factors. So serum is basically that part of blood which is left after clotting of the cells. The clot basically contains fibrin, cells, and the clotting factors. So fetal bovine serum is an amber colored fluid which is derived from the fetal calves post the coagulation of blood, and apart from growth factors it also contains nutrients, enzymes, hormones which are essential for the faster growth of cells, and therefore it is added as a growth supplement in the basal medium used for the in vitro culture of eukaryotic cells. It also contains growth and attachment factors which shall facilitate the binding of the cells to the tissue culture plate. Its composition varies from lot to lot and is usually used at a final concentration of 5 to 10% of the basal media for the culture of the cells. So that's all for today's session. I hope you liked the session and look forward to you to join the next session at the same time. It is next Tuesday 7 to 8 p.m. Thank you, everyone.